Hi everyone, welcome to your lecture on DNA technologies. It is the last lecture in your unit on DNA, RNA, and protein synthesis. Scientists use their knowledge of the structure of DNA and its chemical properties to study and change DNA molecules. Different techniques are used to extract DNA from cells, to cut DNA into smaller pieces, identify the sequence of bases in a DNA molecule, and to make unlimited copies of DNA. Next, we're going to talk about the tools of molecular biology. The first tool is DNA extraction. Biologists use chemicals to remove DNA from the nucleus of the cell. We already did that in class when we removed DNA from strawberries, and you were able to keep some and take it home. So that was the DNA extraction lab. Another thing they can do is cut DNA. DNA is much too long to be analyzed, so we cut it into smaller fragments using restriction enzymes. They cut DNA at a specific sequence of nucleotides, and then the staggered cuts can later be recombined. Another tool of molecular biology is the ability to separate DNA. DNA fragments can be separated by weight and length in order to analyze a specific sequence. One way to do that is through gel electrophoresis. DNA fragments are inserted into a porous gel, and then voltage is passed through and it pulls the DNA to the opposite side of the gel. The DNA sequence can be read because the lighter segments will travel further and faster. So on the picture, if your DNA is inserted into the wells here at the very top of the photo, the larger fragments are going to be closest to the wells, and the farther it goes away from the wells, the lighter and smaller the fragment of DNA is. If you are comparing two samples to each other, the more identical the samples are, the more alike those segments of DNA are. The next tool of molecular biology is the ability to sequence DNA. Once the DNA is in a manageable form, meaning that we've went ahead and cut it to a size that we can handle, it can be sequenced and it can be read, studied, or even sometimes changed or manipulated. And the goal is to eventually be able to reinsert the DNA back into the cell. Transformation is another tool of molecular biology, and a cell takes in DNA from outside the cell, and the external DNA becomes a component of the cell's DNA. One way that the scientists do that is it's called recombinant DNA, and that is DNA that's made by connecting fragments of DNA from different sources. So the first step, and here's your picture here, is to use a restriction enzyme to cut the sequence, and what is left over, if you look at this second photo, based on where it cuts at a very specific sequence of nucleotides, and so that leaves some free nucleotides on either end, and we call those the sticky ends. And that is an area where you can insert a new piece of DNA and add it to that strand. So what's left over? A double-stranded DNA fragment with single-stranded end. So you saw that on the picture on the last page where you had the sticky end, that little bit that was hanging over. So next, a vector is used to insert DNA into the new source. And we talked about vectors during the cell unit. And we said that vectors are an animal that carries disease and gives it to something else, another organism. We talked about bats being a vector. They could, if they bite you, they can give you rabies. We talked about ticks being a vector. If they bite you, they can give you Lyme disease or a number of other things. We also talked about mosquitoes. Here, it's a very similar. The vector is a carrier in which a foreign DNA can be transferred to the host cell. So again, it's a carrier, but a lot of times the vectors aren't other animals. Here they can be like the bacteria or the virus themselves, and we're using it to transfer not a disease, but a segment of DNA. This picture here shows a bacterial cell with a plasmid in it. A plasmid is the circular DNA in bacteria, and the DNA containing the gene for the desired trait is inserted into that bacterial plasmid, and then using restriction enzymes and DNA ligase acts kind of like glue to put things back together. And then that bacterial cell is introduced into the plant cell in a culture, and that bit of information can be inserted into the plant chromosome, and then the plant can be generated with a new trait. So continuing on about vectors, vectors can be either mechanical, where humans are going to introduce DNA into a nucleus, or they can be biological, where we use a virus and plasmids, a small ring of DNA found in the bacterial cell. So they can use viruses or they can use bacterial cells as vectors to carry in there and insert the piece of DNA. This is a picture, again, showing that same process of cutting the DNA at a specific point using a restriction enzyme. In this instance, the restriction enzyme is called EcoR1, and it cuts at this very specific point, CTTAAG. You have to have that sequence, and then it will cut right between the A and the G of that sequence. And after it cuts it, they're able to rejoin the pieces using DNA ligase, which is just another enzyme that acts like tape to join all of them back together. Another tool of molecular biology is PCR, or polymerase chain reaction. And we did have information about this in another PowerPoint. Actually, specifically in your DNA replication PowerPoint is the information about polymerase chain reaction. It's a process by which scientists can copy a segment of DNA, such as a gene, multiple times over and over. So you take just this little chunk of DNA, and by cycling it through different heat and cooling cycle, and having free-floating nucleotides in there and different enzymes in there, they're able to denature the strand of DNA, which is separating it into two separate strands, and put a little primer on there telling it where to start, and extend the strand, and then separate them again, put the start location on there, extend. After 30 times of doing that, you can take like one strand of DNA and end up with over 1 million copies of it. You can get a lot very, very quickly. So in this picture here, you see how many you have after one cycle, then after 2 cycles, and it says 20 or more cycles, you can have over 2 million copies of that chunk of DNA. Okay, manipulating DNA. This is kind of fun. Love the little glowing monkeys in the bottom left. There's different reasons that scientists might want to manipulate DNA. And what we're talking about here is inserting specific genes from other organisms into a different organism. That's why the monkey's glowing. So those are transgenic organisms. One that contains recombinant DNA, or DNA that has been recombined and reinserted in there for a specific purpose. Now, agriculture, industry, medicine, many other fields use transgenic organisms. Sometimes they're referred to as GMOs, but there are other uses. So agriculturally, for profit, you can make cotton and rice that are more resistant to disease. By inserting specific genes, you can help them to grow faster. You can help them just increase their resistance to pesticides, insecticides, all sorts of things. In industry, you can create bacteria that can eat oil, goats that produce clotting proteins, and fish that grow faster. And in medicine, you can create bacteria that are able to produce insulin, which would be very good for diabetics. And other example are glowing tobacco plants and glowing monkeys. Now, a lot of times the reason that they make these glowing animals, this glow gene, is actually very convenient. They'll use it in medical research. So they get the glow gene by taking a gene for bioluminescence from a jellyfish or a coral or some organism that naturally has that glowing factor. And they insert it into, a monkey or a pig or a cat or whatever, a mouse. If you look up like glowing animals, you'll see a whole bunch of them, and they're actually real. They can glow because they've been given that gene. But one reason they may want to do it is that if you want to study a specific gene and see how it impacts or to cure a specific disease, it's very hard to see if that gene went in successfully. But if you put that gene along with the glow gene and insert them both at the same time, then you don't have to sequence the whole DNA strand to see if your gene that you want for a specific disease got put in there. You can just turn on a black light and see if the animal glows. And if it glows, if that glow gene got in there, so did the gene that you're trying to study. So it's an easier way for scientists to see if the gene that they put in there because they want to study to see if it's going to help cure a specific disease or help with a treatment or anything is working, or if it's actually there. And then they can proceed. So to create a transgenic animal, here's the picture with the cow. The gene of choice is manipulated and prepared in a laboratory, and then it's injected into the egg of an animal. And then that egg is implanted into a surrogate mother, and it will grow into a cow that contains that specific gene. The human genome is just a fancy word for all of your DNA and all of your DNA sequence and what it says. So DNA comes from blood, hair, skin, bodily fluids. If you do one of those DNA tests like 23andMe or Ancestry or something like that, they will have you spit into a vial and send off your spit because they're able to get your DNA out of it and they can sequence it. So the goal is one day by studying all of that to be able to provide gene therapy where the bad gene is replaced with a normal gene. And so if you have a gene that is broken somewhere, if we can replace that with a functioning gene, then the hope is that that fixes the problem of whatever's going wrong. And the last thing that we're going to talk about that is a tool of molecular biology or something that can be done with DNA is creating clones. And a clone is a member of a population of genetically identical cells produced from a single cell. So single cell organisms that reproduce asexually are genetic clones already. So bacteria reproduce asexually through binary fission, and they're clones of each other. When your skin cells reproduce through mitosis, they are technically little clones of each other. But to create a clone, to take an organism and make a baby organism that is the exact same, you have to do something a little bit different. So we're going to talk about a sheep named Dolly, and it was the first clone of a multicellular organism, and she was created in 1997 by a guy named Ian Wilmot. Okay, so if you start up in the upper left-hand corner here with sheep A and sheep B, this is how you make a clone. You need a donor cell taken from the sheep's, it was taken from the sheep's udder, but it could be taken from anywhere. You just need a donor cell, one that is not a sex cell, so a body cell, a skin cell or something. And from another egg, or from another cell, you need an egg, okay? So the egg cell is taken from an adult female sheep. Then they remove the nucleus from the donor's like skin cell or something. because it has all of the chromosomes you need, whereas the egg cell only has half. And they remove the nucleus from the egg cell also. The nucleus from the skin cell is then fused into the egg. So now you have an egg that has exactly the amount of chromosomes it needs, and they are all identical to sheep A. So sheep A here would be the sheep that you are cloning. Then you take that egg that has a full set of chromosomes now and stimulate it to begin dividing, and it will create that blastocyst, or that ball of cells that forms an embryo. And then you're going to place that into the uterus of a surrogate mother. There has to be a mother that's willing to, allow that embryo to grow into a baby and birth it. So then that embryo grows and develops into a normal lamb. It was born, and it was a clone of sheep A. And that is how you create a clone. Currently, it is illegal to create a clone of a human. And it is not always successful. There were tons and tons and tons of trials where they tried and failed with creating the clone of sheep. Have a good day.